Welcome to the Manitoba Ag Days podcast, where we hear from some of the most relevant, up-to-date, informational speakers in our industry. In episode 6, Greg Patterson, President A&L Laboratories Inc., shares his story, Healthy, Living Soils Grow Crops.
Thank you. Good morning. I'm going to start off by giving you a little bit of a history of agriculture technology before I segue into where we're at today with soil health and where we see the future going in production agriculture. When we talk about soil health, I coined a phrase that basically we're looking at the new generation in agriculture technology. I call it the evergreen revolution, and you'll understand why as I go through some of these slides. When we look at production agriculture today, what we have to do is we have to focus on what it's going to take to grow the crops that we're looking at, good productive land, quality seeds, water, fertilizer, all those things we know that make it happen. We see production agriculture having a challenge around the world because of the type of ground that we actually have to work with as we push, civilization pushes us on to more marginal land. So we start out with the first green revolution back in the late 1800s, where the Peruvians actually started excavating bird guano from an island and using that for fertilization of croplands. By 1910, they basically had exhausted some of this resource, and it was being regulated and no longer available. Then shortly after the war, we had a lot of munitions laying around. Nitrate nitrogens that were used in the war were now going to be used in agriculture. So that basically started the second green revolution, and we had a whole bunch of things happen at this time with better cultivars of grain, shorter stalk strengths, more use of phosphate fertilizers, and better products to use in production agricultures, and that's kind of where we went into the second green revolution. Well, here we are today, and this individual here, Bennett, actually coins the phrase molecular microecology, the evergreen revolution. And what we're doing here is looking at understanding more about soil health and the relationship between plants and the what I call the microbiome of the plant. So the looming question is here is increasing concern is how we're going to feed the population of the world going forward. With increasing population, the land we have to grow crops on is actually shrinking. Diets worldwide are changing. People want to eat better quality food, more protein, more meat proteins, and again, increased need for higher nutrient density in a food that Jared actually alluded to a little bit here. So we're all focused on better quality food, and we have to come up with how can we do that. So what can we do? Well, one direction has been suggested as a possible reprieve is the need to move more towards ecological agriculture. And why? Because ecological agriculture aims to substantially reduce the dependency on fossil fuels. And that's something we have to be concerned about going forward. So ecological agriculture practices that require greater reliance on natural soil processes, native microorganisms, and the interaction between plants, animals, and humans. And that's what we coin or look at as actual soil health. What are these functions? Well, we see it now, nitrogen fixation, free nutrition, and that's been going on for quite some time with legumes. Biological control, disease suppressive soils, and we'll talk more about that here in a few minutes. Growth promotion and biostimulation of products using some of these products we've identified to promote better growth. And then again, looking at soil community functions, multiple activities that promote growth, suppress disease, increase nutrient use efficiency, all the above. And then how do we manage them? How do we make it happen? So examples of ecological-based agriculture that exist today, rhizobia from microbial fixation, in diverse number of crops. It's been around for a number of years. We're looking at things like the Brazilian sugarcane industry, where they focused on looking at phenotypically responsive endophytes in the soil that recognize that particular variety of sugarcane and produces nitrogen, and there's millions of hectares being grown of this sugarcane presently. And then development of soils that have microorganism populations that enhance plant growth and suppress disease. And that's kind of the direction we're going. So we look at this particular organism here. Rhizobia has been around for a long time. It's basically a Canadian technology. But we identify that this has worked and does work, and we all understand nodulation of any legume and what it does in producing nitrogen. But again, we have a little more to understand here. But why does it work? Because interaction has been maintained between the breeding of these particular plants and a phenotypic relationship with the rhizobium that actually will cultivate these plants. We're constantly trying to find the more aggressive phenotypic relationships, better rhizobium, more productive rhizobium. There's all kinds of rhizobium in soils. Some are cheaters, some are actually parasitic to plants and don't do a lot of good. They rob from the plant, so we're always on the search for these better phenotypes. Again, you really can't see that one. Just page through that. But basically what we're saying here is there's a lot going on that we don't understand. In the case of legumes, something we've completely missed, and it's misdiagnosed on a regular basis, is the relationship to boron and rhizobium. This paper here is not our paper, but it has been identified that with better boron nutrition, we have more aggressive nodulation, more active nitrogen fixation. We know that in our findings as well, that boron is actually used as a signal. The plant uses boron to produce a compound to actually signal for the proper rhizobium that it needs, or the proper phenotype of for that plant. It also needs to understand what this particular organism is and what it's doing in the plant. The plant uses boron and the compounds that boron produces within the plant to actually help the plant identify that this bacteria that's invading the plant is actually a friendly. It's not aggressive, it's not a pathogen, it's actually supporting growth. And without those adequate levels of boron in the plant will actually kill the rhizobium in the plant. We see a lot of that late season and any legume that produced that the nodules die off and their efficiencies aren't there. So we have to understand some of these relationships that's going on. And again, this is just that whole recognition of the rhizobium by the compounds produced by rhizobium. So let's go back to looking at production agriculture in Brazil, where these researchers, these breeders, actually went out and selected varieties of sugarcane that would sustain themselves in production agriculture without a lot of inputs, in this case particularly nitrogen. They had a breeding program that bred for a particular variety of sugarcane that still had the capability of identifying the phenotypic relationship of that endophyte in the soil so it would help actually this crop produce nitrogen. So what happened here is plant selection and breeding was based on little nitrogen use, early research utilization of microbes that supported the plant growth and productivity. So they really focused their whole breeding campaign on that relationship between the plant, the variety of the plant, and its ability to still maintain some phenotypic relationship. And what that amounted to here is, in Brazil, they use about 50 pounds of nitrogen to grow a crop of sugarcane, whereas other places around the world, substantially more. Sugarcane also produced in Brazil contains a phenotype that if that particular sugar or sugar base was used in production agriculture, these bacteria that are even in the sugarcane sap actually suppress disease. So there's some real significant differences between these. Jarrett referred to this individual, Hiltner, in his talk, and we know that Hiltner, in actually 1903, coined the term the rhizosphere. He understood that the rhizosphere of the plant was different than the bulk soil. So why or how was this plant actually able to change the population of the microbes in the rhizosphere from the bulk soil? What was that plant actually doing? We're presently working with the University of Western Ontario on their work done with human biome, looking at the human gut. And we're seeing a lot of similarities between the human gut biome and what we recognize now as the gut of the plant. And that's the rhizosphere of the plant. What we eat, as everybody knows, what we eat actually produces our gut flora. It helps us deal with all kinds of different things. And we're finding out now that what the plant eats, what the plant is able to produce in compounds, basically makes up this rhizosphere microbe. So since 1903, we haven't done much. We really haven't tried to understand this in any great detail. And it's been said, it can be suggested that mankind knows more about the vast expanses of our outer space than about the workings of 15 centimeters of soil that support most of the life on this planet. And that's so true. We're starting to go there now with the new technology we're going into now. So when you think about the concept of photosynthesis and what the plant is actually doing, it produces and converts a lot of sunlight energy into compounds it uses in its overall energy drive. Anywhere from 40 to 60% of the photosynthates a plant uses actually ends up and is redeposited in the rhizosphere to feed these organisms. So that's a lot of energy a plant is putting out there to basically deal with this relationship. These are not by mistake. They're very sophisticated compounds. And the plant actually regulates these compounds and selects certain compounds to do certain things, to signal for certain organisms in a soil to do certain things in its whole life cycle. And again, a little hard to read some of these slides, but this is a page out of a document that just shows you a whole list of different compounds that plants give off into the root system. And again, it's no mistake. These are very complicated chemistry that this plant actually develops. In my career, I've been working at this whole concept of soil relationships with plant nutrition and plant physiology. And this individual here is George Lazarsovitz. He worked for the Federal Research Station in London for his whole career, looking at soil microbiomes and soil amendments that stimulate the biome and stimulate growth and suppress disease. So whenever I had a question, I'd go over and him and I would sit and talk. And we'd talk about this technology. And we looked at some of the things he was doing and some of the things that were coming out that were innovative and the technologies used to look at this whole concept. For those who don't know George, he's the one on the left. So he has a lot of knowledge, and him and I said, when are we going to take this whole technology to the industry? When are we going to start using this and applying it? And he said, well, Greg, pretty soon we'll have the technology that will be available to actually analyze this, and then we'll take that to task. Well, at that time, he was working with Sean Hemingson at the University of Saskatchewan, and they were looking at fingerprinting soil types from around the world. Their goal was to go out and select soils from all different parts of the planet and come up with a recognizable fingerprint for all these soils. What they thought they would be able to find, or what they would find, was that a soil from Saskatchewan would be different than Ontario, different from Quebec, different than Amazon, and so on. In reality, what they found out was all soils were the same in the microbes that they contained. Different concentrations and different arrays, but every soil had the same group of bugs in that soil. So that asks the question, well, if that's the case, why is there so much difference in how crops produce or how crops perform? We did some other work here. This is some work I did with George before he came to work with me. And this is looking at scab in potatoes. And how could we suppress scab by understanding these organisms? And this is coming from some of the work that him and Sean put together. This is the organisms in bulk soil. You can see there's this is our research plot in Ontario versus PEI on potato production. That's the bulk soil. That's the rhizosphere soil. Quite a bit of difference. Same fields, same general location, just going from bulk soil out in the middle of the road to what the rhizosphere soil had. And you can see a significant difference. We took it one step further, and we looked at what are the bugs or what are the endophytes the plant's actually taking up. And at this time, this was really unknown, but what we found out here is, I don't know if you can read that back there, those are rhizobium. What the heck is a rhizobium doing in a potato? Well, what we're finding out today is rhizobium and rhizobia type organisms are in all plants. Some don't produce nodules, but they are a very aggressive endophyte, and they do a whole bunch of things in a plant. So rhizobium and disease suppression, they have a lot to do with disease suppression. Again, these are, and you can't read this, but it's basically what it's listing here is a whole bunch of diseases and insects and other disorders controlled in a whole group of plants by rhizobium that don't produce nodules. These are endophytes that the plant actually takes up. Rhizobium influence soil structure. They give off a polysaccharide that is responsible for the aggregation of a lot of soils we look at. So we talk about soil structure and soil quality. Aggregate stability is a big part of that. Rhizobium create that. They put this glue effect on the root system that basically sticks the soil particles to the plant. They are also compounds that hold a lot of water, so improve water use efficiency in soils with proper endophytes, rhizobium endophytes, and a whole bunch of other things that they do. Again, I don't know how you see this, but this is a root system. This is courtesy of Michael Dolinsky, if anybody knows him. He took this picture of a barley root, and this is just basically all the soil particles adhering to that barley root because of this compound that rhizobium create and give off at the root system. Same sort of thing here. So what these rhizobium do is they produce compounds that, one, stick the soil particles to their tiny little root hairs. They aggregate the soils, give it better porosity, better water holding capacity. And actually, every bug that is on this planet, and even in our own bodies, main goal in life is to help us convert phosphates to a form of energy that we use to live. Every plant uses the same thing. This production of ATP phosphate from inorganic phosphorus is what drives us all. So these organisms we're analyzing, their main goal in life is to help the plant take up phosphorus. Because without phosphorus, nothing's happening. So they're great solubilizers of phosphorus as well. So our phosphorus efficiencies will increase with better microbial activity. This is just looking at profiles of different organisms we found in those two soils in Ontario and in PEI, and showing the relative differences. Our Ontario fields were very much more productive, had less disease, less scab pressure, and that begs the question, why? Same fertility type practices, what's the difference between the soils? Well, we found out in Ontario that we had a greater proportion of the good guys than the bad guys in Ontario versus in PEI soils. So the concentration of what we have now identified are the organisms that help the plant be more productive were greater in the higher producing areas than in Ontario than the PEI, and we had much lower scab occurrence in Ontario than we did PEI. And this is just a chart in a table looking at the relationship to the nutrient balances and what we were looking at that actually helped us control scab in these potato fields. So we do have and have now a cultural technique, I guess, if you will, on dealing with potato production and reducing scab or controlling scab. And that particular information is being used worldwide, and it was published back in 2007 in this journal. So we're getting a better understanding of what we can do to make us more productive and reduce some of the issues we have in production agriculture, looking at these relationships between the organisms in the soil, and what can we do as growers to stimulate these bugs to actually occupy the rhizosphere and actually be taken up by the plant. So in 2010, George Lazarowicz retired from the Ag Research Station in London. He now works for me. We started a company called A&L Biologicals. And since that time, our main focus has been looking at just what is the relationship to plants and these different organisms, what are the bacteria, what are the fungi doing, and how can we basically control that. So our main goal here is, what are these bugs? How do we identify them? How do they populate the host? How does the host select for these microbes? In other words, how do they get from the bulk soil into the rhizosphere, and how does the plant actually populate that rhizosphere? How is the host responding to them? What are the unique characteristics of each of these individuals? So we not only looked at the bug, we looked at the metabolites they gave off, and what did that metabolite do in the physiology of the plant to make things different. So we're looking at all this. So what are these bugs doing? And then what are the forces that maintains this equilibrium? How can we actually create this better rhizosphere, and how can we keep it maintained? Because that's what's given us the response. We had the luck, I guess, if you will, of working with this individual. He was a farmer in southwestern Ontario, a little bit outside the pack. He thought outside the box, and he came up with a strategy on how to produce a better crop of corn. He was growing over 300 bushel of corn, and all his neighbors were, they were struggling to get 150 bushel. So significant difference between what he was doing and what they were doing. He worked with minimum till or reduced till. This is kind of his planting pattern between corn and beans. He had controlled traffic, so he wasn't compacting his soils. And I asked him, I said, Dean, how long does it take you to get from that type of production system of a brand new field to where you are today? He said, it takes about 5 years to change that whole paradigm on a farm. And that made sense with what he was doing. So, first year into this whole process, and George and I have known each other a long time. He's a plant pathologist, and I'm a physiologist, so we've agreed to disagree for quite some time. I'm a firm believer that through nutrition we can help the plant fight disease. He thinks disease is disease. You can't do that. It's just a disease. But what he did notice first year out with this work we did on Dean Glennie's farm is there was a definite difference in the microbial population in that rhizosphere when we looked at just pH. So he is already seeing some relationships to nutrient availability, or particularly this pH relationship. So I don't know how well you can see this. I'll try to explain it. So everybody knows that we talk a lot about diverse population of bugs in soils is what we're looking for. Well, diversity is great, but that's just where we start. So we look at the difference between that 150 bushel crop and Dean Glennie's 300 bushel crop. And if you could see this scatter diagram here, this is a sample of the soils of both of those fields in the early beginning. So looking at, before planting, what the population was in the bulk soil. Then shortly after planting, we take an analysis of the rhizosphere, so very tiny plants. And again, we see a very, very diverse population and really no significant difference between the two. Okay, so we got this big, diverse population of bugs. However, by day 30, as we start looking up inside the plant and what's going on in that rhizosphere, we start seeing some differences. And we're using a process called TRFLP that basically groups communities of bugs into specific communities based on who they are and what kind of genes they possess. By day 60, so that's what we're seeing at day 30, but by day 60, we see a real significant difference. This is the 150 bushel crop in its population, very diverse population, and this is Glennie's field, again, completely unique, but now we're getting to get a more unique population. We don't see everybody in the hood anymore. We see a very selective group of bugs that are entering into that rhizosphere. So a real shift in who these bugs are. So the plant is doing something in the one field that's looking at this diverse population of these organisms in the bulk soil and saying, I want you, and you guys take off. So I'm only prepared to let you guys sit at the table. I'm only prepared to feed you guys because I know what you guys can do for me. Now, in this field, the plant doesn't have the power to do that selective. It can't feed enough of the population. It can't put enough on the table to bring the good guys into the hood. So it's not doing that. So we see a real separation. We see that right through to 90 days. After the plant starts to senesce, we see the population go back to a diverse population. So as the plant starts to senesce, no longer can the plant fill the table with the good groceries. It goes back to just a free-for-all. So we see the diverse population start to come back into the rhizosphere. When we look at the chemistry of these fields, I said in our research work, we analyze everything to death. We look at soil chemistry, we look at herbicide residue, we do all this to understand what's going on. We notice that in Glennie's field, he has a very good balance in nutrition. This goes back to our scab research, knowing that this number means something in controlling scab. And when we compare that field to the other field, there's a significant difference in that whole balance in nutrition, and particularly between potassium and magnesium. Potassium and magnesium help any plant, anything in our body, convert phosphorus to ATP. So there's a direct connection on why these two compounds particularly are very important. The other thing we keep looking at in soil health is the Solvita test. Anybody use the Solvita Verse test or know what it's all about? Well, it's a test you can do on your farm, or you can send it to us. And through this simple test, we can measure the CO2 production or the respiration rate of the bugs in your field. And it's understood that with higher organic matter, you should have better biological activity and better bugs and all that good stuff. Well, I've had a lot of customers call us up because we're the distributor of this kit, say, does this thing go stale? Does it run out of power? Could there be something wrong with it if it's been on the shelf too long? I said, not that I'm aware of. I said, why do you ask? He said, well, I got some kits from you, and I tested my high organic matter soil and my sand soil, and the sand soil has better respiration rate than the higher organic clay soil. Well, at that time, I didn't understand that. But because we're doing this type of testing, and we're looking at all these different soils we're working with, again, this is Hessels versus Glennie's. Glennie's field has an organic matter of 2.9 to 3%, and its respiration rate here is at 67. Hessels has 7.8% organic matter, respiration rate at 59. Another part of the field, Hessels 6.8, organic matter 63. Glennie's at 2.9, and he's at, I think that's 76. So organic matter doesn't make a difference, according to this. Goes against what we all believe. Everybody's belief is the higher the organic matter, the healthier your field is, the more productive it is. Well, it's not what we're seeing. So let's take this concept back a little bit and say, okay, in theory, if plants can do this, why has it happened? Why can't plants forage for themselves and give off these compounds to do this. Well, in theory, that is happening. Weeds do that today. Weeds can grow on a rock. They've evolved over time. We haven't bred out the phenotypic relationship between what they are and what they select for, or these bugs that they use. They do that now, okay? So we've actually destroyed the plant's ability through our breeding efforts and not paying attention to these possible phenotypic relationships between plant varieties and these bugs. That just shows you basically some of the differences in micronutrient uptake between a typical weed and some of the plants we grow. So it's a real phenomenon, and it's something we see. So what we're understanding here is soil health is about the chemistry, how this plant uses nutrients and how it is able to give up certain compounds to select for the right guys, clean up the hood, if you will, and only let the right people in the neighborhood. We see that potassium has a huge role in this signaling process. It's one of the main components we see in the whole signal process, particularly when it comes to disease-suppressive soils. So our research takes us to the field, and we look at the spatial variability within all kinds of fields across southwest Ontario, and again here in a few locations in the west. We go with our drones, and we fly over these fields, and we look at the NDVIs of these fields, and we select areas in those fields where we have very high productivity, marginal productivity, and low productivity across a number of fields. In this case, we're up to about 50 fields we're working with at the present time. So we take multiple samples in this field based on this criterion. From there, we start looking at what are the populations of the organisms. We use all kinds of PCR techniques and qPCR, looking at genetic compounds. And we start comparing things like Pseudomonas and Bacillus and Rhizobium and nitrogen fixers and understanding what they do. So we look at, in this particular case, quantification of functional genes in roots. So we're looking at the functional genes. We identify what genes we're producing. These bugs are genes these bugs have producing certain compounds in both the good producing and bad producing fields. These are actually the genetic markers for nitrogen fixation. So we see that, and we can quantify that. We look at yield versus one of the indicators we have in one of the algorithms we've produced called our General Fertility Index. And we see that there's a direct correlation to our GFI and yield in these fields. We're looking at yield versus K to magnesium ratio, which I talked about already. Yield versus percent K, yield versus aluminum, to try to get some kind of understanding of what these nutrients are doing. And then we look at things like nitrogen, yield versus soil nitrate nitrogen and its relationship, yield versus K. This is another one we looked at here, looking at organic matter. And in our studies, again, a surprise to us, absolutely no yield correlation to organic matter. That surprise everybody? Okay. The available organic matter we put on the soil test, not trash. But we do have another test in our soil health test called our soil active carbon test, which is a measurement of what we believe is the carbons that are actually directly feeding the microbes. So we have that on a test, and we do see some relationship to yield in that test as well. And then we look at that slide again, looking at active carbon versus organic matter. Because we participate in kind of a global soil health group, we asked the question to other labs that are doing some work, and one particular group of labs using Solvita test are looking at the same thing, and that's their relationship to organic matter and yields. So again, it's not just us saying that. There's multiple labs putting this information together saying that, no, organic matter alone is not an indicator of soil health. So now we come up with kind of a synopsis of where we're at and what we understand. And we look at total plate count in a field. I don't know if you can read this, but our general fertility index of the rhizosphere, anything over 0.5 is very significant here. So 0.678, percent K 0.774, nitrate nitrogen 0.488, bulk soil, GFI, which was surprised us, but even the bulk soil in the good fields we see almost 0.6. Here is a phosphorus reading, 0.751 to phosphates. Now, this was a bit of a surprise. As I said, all these bugs, their main drive in life is to solubilize phosphates. But we do see a response in one particular group of bugs to phosphate levels in soils, and that's Pseudomonas. It is a phosphate solubilizer, but the reason we believe that it actually responds to better phosphate soil levels is it's a bit of a wimp. a lot of things can wipe out Pseudomonas. So it likes a little better living condition to make it work functionally. And that's where we see that its drive and its ability to solubilize phosphate comes from having a little more phosphate in the table. So we see that, and that's the only place we see the correlation to phosphates. And this K to magnesium ratio, we see correlation across all of them. So we start adding up some of these things. We look at Pseudomonas itself, and again, we're looking at the rhizosphere, GFI 0.95. Again, it really likes a full table. So it really responds to fertility in general. Percent saturation, again 0.9. And here's where we see the saturation of phosphorus. Nitrogen to sulfur is 0.8, and even some for chlorides. So Pseudomonas, as I said, is a bit of a wimp, so it likes things just a little better condition. A little more TLC for Pseudomonas. Rhizobium, we see, again, the GFI in the rhizosphere at almost 0.7, base saturation at 0.64, nitrate nitrogen 0.76, bulk GFI at 0.64, soluble salts, nitrate nitrogen, K to magnesium ratio, again, at 0.6. Here's where we see the real significance with boron. And again, it's a rhizobium. And we've got other literature that says that nodulating rhizobium respond to boron, but in this particular case, our endophytic rhizobium also very strong correlation to soil available boron. So we take this relationship, and you can't see this again very well. This is some papers done, not by us, this is some work done by researchers looking at mangrove groves in other parts of the world and just what makes their soil biota in better shape. And they're actually finding out as well that nutrient concentration, and in the case of the mango grove, the particular bugs that support the mango are very sensitive to boron. So again, another response there. And I don't spend too much time with those because you can't read them. So I asked the question to my technicians. I said, you know what? When I look at these profiles and I look at what I would expect to Pseudomonas and Bacillus and Rhizobium, because when we look at the numbers now, we can almost see the predictive bugs that will be there based on some of these algorithms. And I said, you know, I don't really agree with some of this data because the Rhizobium in this particular field, based on the bulk GFI and the K and everything we see, should be a lot stronger. But it's good, but it's not where it should be. So I had them go back to the data and select out the data going from the lowest boron to the highest boron, and look what we found. Almost a straight line, linear correlation to boron levels, even though it's all low. And this is why the technician said, I don't know how you want me to look at that, because all of our boron levels in our trials were low, but this is what they found. So again, very strong correlation to what we're seeing in the data set and for Rhizobium and this relationship to soluble boron. So we look at the different bugs. We know the gram negatives are the good guys. The Pseudomonas and Rhizobium are really the guys doing the job. The Bacillus are gram positives, and they actually have a bit of a negative impact on performance. Now, we have a lot of products in the marketplace that are basically Bacillus. The reason we're going there is Bacillus are tough and rough, and you can't kill them. So they're very stable in anything we put them on, and they do respond. They do actually give us some stimulation. Matter of fact, when I look at our soil health index and we see the population of the bugs move from a very diverse population to start being selective, the first one I see at the table is Bacillus. So as the plant identifies it's starting to get a better rich environment, it starts moving Bacillus in. Bacillus are crazy antibiotic producers, and this is why they work in the products we're putting on with. So the plant, my thinking is the plant says, well, things are getting better. I want to clean up the neighborhood a little bit, so let's bring in the antibiotics and clean things up. They're the paramedics, if you will. Bring them in, clean things up. And then what we see as the soil health index moves from, you know, just the low, medium level up into the good range, the Bacillus population drops from 10 to the 6th to 10 to the 4th and maintains there, and our Rhizobium and Pseudomonas and nitrogen fixtures go up. So there's some real shifts happening in this rhizosphere that we're seeing happening from these different communities. So again, the gram negatives versus GFI, a strong correlation, and the gram positives, the opposite. Here we have yield versus GFI and low gram negatives. So the lower the gram negatives, the better the yield. The higher the yield versus GFI with high gram negatives, again, very strong correlation. So we start putting this stuff together, and we start seeing some real neat things from some of the data and some real predictives. So microbial diversity, when we talk about overall microbial diversity, The complexity of interactions between plant and microbial communities increases with microbial diversity. The resulting diversity and complexity may act as a buffer between the positive and negative effects. In other words, a diverse population is not necessarily a good thing because the plant can't feed everybody. It's when we get a good population of bugs that the plant can actually start selecting from that we have a healthy soil and an active environment. Nutrient availability determines endophytic survival. Endophytes prefer specific carbon sources. As I said before, the plant produces certain compounds to feed the bugs it's selecting for. Competition for these nutrients can be a determining factor in shaping the endophytic community within the plant. Specific nutrient availability may limit the number of endophytic microbes that can be taken up and survive inside the plant. So, again, understanding that relationship is where we're at. How many people in here work with green manures? Green manures work? Absolutely. What's the best one? Well, if you read this guy's book on this green manure, it's the best. You read this guy's book, it's the best. Green manures work, and they work. Why? Well, we grow a crop, and we have the crop in the field for maybe 4 or 5 months, feeding these bugs. Then we take the crop off, there's no more food. You put a green manure in, you now have a green living plant returning nutrients to the rhizosphere to keep these things alive. So green manures, no matter what green manure you use, will help maintain that population that you've started to build. If you're doing it right, you bring in a green manure and put it in there after you've harvested that can overwinter and cover that soil. You've got a food source for those crops. So green manures work, and I don't care which one you choose. You know, we do have good information that some rotations are different than others, but any green manure is powerful. So what have we learned? We've learned that soil health is all about the chemistry. The chemistry we feed the plant and what the plant feeds the rhizosphere. And we now have those compounds, that's what we talked about. So we focus on nitrogen fixers, rhizobium, Pseudomonas, the gram-positives. There are some good bacillus, but for the most part they are negative. And we try to build profiles to understand who they are and how we can stimulate that whole reactivity. And again, we use NDVIs. When we select these fields, we have it correlated to biomass and yield in the field, and we're starting to look at how those NDVIs actually correlate to some of the other nutrients and some of the other parameters we're looking at. So this is a predictive model we have. The one line is the actual yields and production. The other line is a predictive model. So this algorithm we've produced is very, very accurate. I mean, its R-squared is pretty phenomenal. Now, when you look at what we've done here, we have looked at these relationships. We've looked at the relationships of the types of genes through qPCR, looking at the genes that we're looking for. We're looking at the organisms themselves and trying to predict who they were and what they're doing. Matter of fact, within this predictive model and within this, based on some of these high peaks, we actually are recognizing a disease-suppressive soil. So we're getting there. We've got a lot of stuff we don't know yet, but we've got a fairly decent predictive model to help you understand what's going on in your field. And not only that, when you do this soil health test, again, you can't see too much of this, but if your soil health index is on the low side, you look to these graphs and charts and tables. If one of these things are outside the optimum range, you work on that, your soil health index is going to go up. And based on the numbers we have and what we have done over the last 8 years, we actually know that based on certain things, we can move into that rhizosphere a certain organism. We know what the bacillus respond to. We know what the rhizobium respond to. We know what the Pseudomonas respond to. And without even going out and plating it, we can almost predict who's going to be at the table based on where this number is going to be. So that's what the soil health index looks like. We also look at the chemistry, residual chemistry. There's a lot of information out there that suggests that the compounds we're using in production agriculture are very detrimental to the health of our plants and the health of us as human beings, right? We hear it all the time. This is a good news, bad news story. If your soil health index is here, even if you have a high residual chemistry, it doesn't matter. There's only about 2 groups of compounds that are affecting these organisms, but they don't have any impact whatsoever if you have a high soil health index. These bugs are resilient enough that they can withstand anything we throw at them based on what we've seen in our soil work. So that's a good news story because we need these products. We need to be able to use these compounds to help us in production agriculture. And again, on this report, it's hard to see, we give the total respiration rate with our soil health, potential mineralizable nitrogen, and basically the biological quality of that particular soil. And that's actually showing a poor soil health test, but it's hard to read from there. So if you look at what we do, we have a whole suite of soil health tests. This is our basic soil health test. Residual chemistry test is not part of this. It's an optional test, and it's quite expensive. Most people don't actually go there because we're convinced them they don't need to. But again, we do pretty much everything. We do aggregate stability. We do SAP analysis. We do biological testing, a whole array of different things. So in summary, balanced fertility provides consistent yields and quality, while at the same time builds a more sustainable ecological soil, or healthier soil that we've talked about here. If you want to look at more of this stuff and look at where we come from and what we're doing, you can go to our website and you can download some of these newsletters that talk about where we're at and what we're doing from April through to July. If you go to this particular place on our website under soil and download these two tech bulletins, these two tech bulletins actually talk about what I've talked about this morning, give you a better understanding what soil health is all about and where we see it going in the near future. So pretty quick analysis of what we're doing today, but I assure you that this information is fairly new. We do see the correlation in it being strong, and it does give us a lot of information on how to be more productive. One thing, I'll go back to Dean Glennie's comments. I use 1.5 pounds of nitrogen to grow a bushel of corn. For most production agriculture, that one field that grew 150 bushel, if you looked at the relationship of nitrogen use, that's what we would use there. Dean Glennie's at 0.9. Our nutrient use efficiency is going to go up. Our water use efficiency is going to go up with these healthier soils. So as we start supporting the plant, so the plant can support itself to actually sequester some of these good bugs into the rhizosphere, our nutrient use efficiency, our overall productivity will increase. So going forward in the future, we'll be more focused on this just to do a better job with what we've got to work with and just be more efficient on the farm. So with that, I'll cut it off there. I think I still have a little bit of time. Yes? Our fertilizer recommendations, when you do a soil health test, we usually have you send it in with a regular soil test, and you get the recommendations on the regular soil test. So it's part of it. No, we don't, but most people that are doing a soil health test are doing a regular soil test with it. So that's where you get the recommendations. So soil health is kind of set up to give you more a description of what we expect to see in rhizobial, or the rhizosphere community. Yes?
Just repeat the question if you can.
Okay, the question was, most farmers use muriate of potash as a potash source, and what kind of impact it has. Is one better than the other? First of all, muriate of potash has a higher salt index, so that can be a problem, but these bugs are very resilient to that type of salt. Roots aren't, but the bugs are fairly resilient. And the chlorides in potassium muriate of potash is not chlorine, so it's not that nasty gas we've been told. It's actually an essential nutrient for plants. When I look at a sulfate source of potash versus a muriate of potash, sulfate sources of potash are taken up by the plant much quicker than a muriate because of the sulfate ion and its reaction at the root system. So other than nutrient uptake directly to the plant, there's really no difference. Muriate of potash is less expensive. Broadcasted out there, it's not going to have any difference than a sulfate source. If you're using potash in season because you have low potash availability and want to increase your potash availability, sulfate sources are better. So they're worth the extra dollar in season for planter mixes, close to crop growth, that type of thing. Yes, the microbes help solubilize all that. We have microbes that solubilize potash. We got microbes that solubilize zinc. We got microbes that solubilize phosphates. Their goal in life is to solubilize nutrients that the plant needs. And the plant will actually sequester those bugs. What you have to do is look at where that level is. Because when we look at availability, too high a potash is just as bad as not enough potash. The plant will actually shut down from taking up potash once you get to a certain range. And you need to know what you can do to that soil to bring that back in range. It might be just simply drawdown. It might be the addition of calcium products to change that relationship. But when we look at potash, don't focus on parts per million. You want to focus on the exchangeable amount of potassium available to the plant. So when we look at your percent available potassium or your percent available magnesium or calcium, what we're trying to reflect on is the coefficient of diffusion. In other words, how readily available is that plant or how quickly that soil is going to give up that. You might have a soil that's high in CEC that might have 300 ppm of potash in it, but on the exchange complex it's less than 1%. The higher the clay content, the higher CEC. What that number is telling you is, yes, you've got lots there, but the soil's ability, because it has such a strong buffering capability, to release that in season is not there. So understanding that balance and that relationship is really critical when you look at nutrient availability. We have a new test we've started doing. It's not new, it's been around for a while, but it's based on some research we've done, where we actually look at percent P as well. Now, it's not the same thing. It's not percentage on the exchange site. It's percent availability. And you could have 20 or 30 ppm of phosphates in soils and virtually have none available to the plant because of competition with calcium and aluminum and iron. So when we look at a soil test, what I'm trying to explain to you is don't focus so much on parts per million. All soils are not the same. How many people in here agree with me sands and clays are different? Pretty simple, right? So the mineral components of those soils are different. Their optimum capabilities and where they should be to satisfy plant requirements are different. And understanding that relationship so you understand where the plant availability exists. Now, when you look at overall coefficient of diffusion, water plays a role. So the higher the water content or water availability, the lower your coefficient of diffusion needs to be of that nutrient and still be a plant available. But the droughtier soils are, the higher the coefficient of diffusion needs to be to make it available to the plant. And that's still, again, just looking at percent saturation of those elements. I hope I didn't confuse people, but we got to start focusing on overall availability rather than just how much is there. No, that water soluble test is something completely different. It deals with soil structures, it deals with compaction, a whole bunch of different things.
We could take another question if
Yes.
there is one.
You're not going to improve your biome by growing more legumes. You'll improve the quality of your field by having a growing crop on it more time through the season, be it a legume or be it a rye crop, some green manure. Your overall soil health is going to improve as you improve the overall nutrition to the plant so the plant can return the carbon source to feed these bugs. And that takes about 5 years to get going in the right direction. Your soils all have the right population of bugs in them to get to that without any inoculation of an outside product. These biologicals that are coming on the market today are just a helping step to get there. So if I got a poor productive field, if I put on a biological, I'll get a response. But if I don't put a fertility package behind it to help the plant feed those bugs I just put on, they're going to die off. So I can do it naturally. It take me longer. But if I buy a product, bugs in the jug, if you will, and I put it on, and I understand the concepts I've just talked about on having the plant have enough nutrients that it can feed the biome, you'll get there faster. You follow me? So green manures have something there sustaining the sugar for us, the carbon source going back to the population. I want to build that population and continue to bring it up. What we didn't believe is we didn't think that we'd see a a significant response from the bulk soil. But our bulk soils in a field that's healthier to start with are significantly different than a poor field. So we do see some relationships. And in a healthier field, our bulk soils are, again, the right population is there. You don't see it as much as the diversity changes after the crop's gone. But the field's ability to rebound once we put a crop in there and the plant be able to select for this community is faster in these fields where we've started to build that population. I hope I didn't confuse everything there, but as we go forward, we understand what we need to get that population built up. It continually gets better. So our reservoir of the good bugs gets bigger. The plant's ability to sequester the right bugs into the rhizosphere is greater. Okay? What's that? Not necessarily. Not necessarily. We actually, in Dean Glennie's case, he's been no-till for a long, long time, and he's got great productivity. I mean, things are really, his field's number one in our research. We convinced him to plow up one field. I thought he was going to have a heart attack, but he did it. And we actually saw a yield response. So reduced till, I mean, it's better conservation of soils and all that good stuff goes with it, but it's not necessarily an indicator of soil health. Okay? Well, thank you for your attention. I hope I shed some light on this whole soil health issue, and it's not about pennies decomposing in soils.
If we could just share our thanks with Greg for spending some time with us. Thank you very much.
Thanks for tuning in to the Manitoba Ag Days podcast. We'll see you next year from January 22nd to 24th, 2019.